Hello, my name is Alexa Murray, and I am currently a fifth-year PhD candidate in the Joint Graduate Program in Toxicology, working in the lab of Dr. Deborah Laskin at Rutgers University. Most recently, I have been working on a project that involves the use of non-invasive lung imaging techniques to predict toxicant-induced alterations in lung structure and function. One of the primary focuses in our lab are macrophages. Macrophages are phagocytic cells and key players in the innate immune response. Their major functions include protection against viruses, bacteria, and also mediation of inflammatory responses. There are two major subpopulations of macrophages: M1 pro-inflammatory macrophages and M2 anti-inflammatory macrophages. The balance of these two is essential for maintaining homeostasis. Pro-inflammatory macrophages are the first responders. If you're familiar with Star Wars, pro-inflammatory macrophages act as soldiers of the dark side. They release negative energy that stimulates activation of many other cells, cytotoxicity, and tissue injury. Alternatively, when anti-inflammatory macrophages become activated, they act as soldiers of the Jedi Order. These macrophages release positive, protective energy, which plays a role in immune suppression and wound repair. However, if the balance is tipped in either direction, it can result in acute injury or fibrosis or cancer. The balance between the dark side, pro-inflammatory macrophages, and the Jedi Order, anti-inflammatory macrophages, is crucial to regulating inflammation and wound repair. You can find macrophages in many different tissues in the body. However, my research is focused on macrophages in the lung and mechanisms that regulate macrophage activation following injury. The model of lung injury that we use is nitrogen mustard. Nitrogen mustard was originally developed as a chemical warfare agent. Exposure to nitrogen mustard causes acute lung injury, which progresses to fibrosis and is the major cause of death associated with nitrogen mustard exposure. To evaluate nitrogen mustard exposure and macrophage activation, we've created a rodent model where we intratracheally administer saline, which serves as our control, or nitrogen mustard. This research is important because findings may provide novel insight into mechanisms regulating macrophage activation following nitrogen mustard. And if we can better understand the mechanisms, we can potentially intervene with therapeutics. This is critical since currently no treatment options are available for pulmonary injury and fibrosis caused by mustard exposure. We evaluate nitrogen mustard exposure using numerous experimental approaches. We collect lung tissue for pathological examination. We also assess pulmonary function in the airways and lung tissue. And although these approaches are useful, they are terminal procedures and limit the dynamic changes over time associated with nitrogen mustard exposure. More recently, we've implemented the use of two non-invasive molecular imaging techniques that allow us to track the progression of injury and response to therapeutics over time in live animals. The first technique is magnetic resonance imaging, or MRI. With this technique, we can quantify acute lung injury, such as inflammation and edema, and more chronic disease progression, such as fibrosis or cancer. The second technique is computed tomography, or CT. With this technique, we can quantify total lung volume and evaluate alterations in tissue densities that correspond to different components of the lung, such as air, tissue, or consolidation. The overall goal of the study was to develop a mathematical model that would allow for us to use in vivo animal imaging to predict lung structure and function. Creating this model would reduce the number of animals used in experiments and also allow us to apply modeling to evaluate additional pulmonary toxicants. We could additionally use this methodology to test the efficacy of therapeutics. So how did we go about creating this model? We integrated different components derived from our imaging data into a mathematical model. Essentially, the air and consolidated components from rat lung CT scans and volume of lung injury derived from MRI images served as variables in a mathematical equation that would allow for us to predict lung structure and function. In summary, non-invasive imaging can track and characterize acute lung injury and the progression of diseases such as fibrosis. We have known functional lung measurements and pathology that we've acquired over the years and from the same animals that we've imaged. We've been able to compare that data to the predictions made by the mathematical model using parameters we've extracted from the imaging data, which means we've tested many different mathematical models to come up with one that allows us to use imaging data that generates continuous data to make predictions about lung structure and function following toxicant exposure. Thank you.